I'm really happy to be here again to speak to the UAB audience. Today my talk is going to be a little different. So last time I had the privilege to talk to you, I talked specifically about my ovarian research program. And this time what I wanted to do was talk about some of the work I do with students. So I've actually aimed this talk predominantly for students. This would be more of a talk somewhat that I would give in a class, but that would be much more interactive. I didn't know what to expect here, so it's not going to be interactive, but I was really trying to communicate, or what I'd like to talk about are different projects I do with students, because to me, students are really an important part of my research program. In fact, I consider them the heart of my research program. I really love working with students because, for many reasons, one of which is I really like to be challenged and I like to think differently and understand different perspectives. And it brings me great satisfaction to pass the love I have of my work and my research and my knowledge to the next generation of scientists. So my hope today is that my talk is not only going to raise hopefully some interesting scientific questions and ideas, but I'd like it to open the door for continued dialogue with UAB about the educational aspects of your programs. And I predominantly do molecular epi, but for today, I'm just going to focus mostly on what I call more traditional epidemiology, although I'll sneak some molecular epi in there. So again, the unifying theme of my talk will be ovarian cancer. And the first part, I'm going to provide some background on the epidemiology of the disease so that we're all on the same page. And then I'm going to discuss two projects that represent leveraging Consortia to do some interesting work with students. And then like I said, I'll sneak my molecular epi in there at the end if there's time. So the goal of ovarian cancer research is to explain how we get from here, which are the ovaries are about the size of almonds, to here, which this is ovarian cancer. This represents a typical ovarian cancer that by the time it's diagnosed is this large, bulky disease that's really overtaken the entire peritoneal cavity. So as epidemiologists, we're going to be looking at the population level to identify associations between lifestyle, behavioral, or genetic factors in ovarian cancer, with the hopes that we could provide some insight back to laboratory scientists on possible mechanisms of development, which hopefully will open the door to prevention and early detection strategies. We also like to provide this information back for policymaking, as well as direct recommendations for prevention to women. Now, the good news is that ovarian cancer is relatively rare. We only see about 22,000 cases a year here in the United States. Just to give you some perspective, it's about 1/10 as common as breast cancer. So whereas 1 in 8 women will develop breast cancer in her lifetime, only about 1 in 70 to 80 will develop ovarian cancer. And as you can see from the American Cancer Society facts and figures, it's not even in the top 10 cancers in women. It ranks number 17th overall. Unfortunately, it's actually a very fatal cancer. It's the 5th leading cause of cancer death in women, and it's the most fatal gynecologic malignancy. So let's talk for a minute about incidence and death rates. First, as you can see from these graphs, ovarian cancer has very significant geographical and regional differences. And just out of curiosity, I just wanted to point out, there's Alabama compared to where I'm here in Pennsylvania. We're not doing as well incidence-wise, age-adjusted as the country, and we're also not doing quite as well as Alabama. So you can see very differences in incidence and also in death rates. So in addition to these regional differences, there are also substantial racial and ethnic differences in both incidence and survival. But it's very challenging to study ovarian cancer in general because it's a rare disease. But studying it in non-white women is extremely challenging because it's so rare and the incidence rates are lower. But as you can see, especially for Black women, the incidence rate is low, but the death rates are higher than compared to the other ethnic and racial groups. So again, there's progress that we need to make in studying these different populations, but it's challenging because of the rarity of the disease. So in terms of survival in general, we've made progress over the last 40 years, and that was really due to the introduction of platinum-based therapies in the 90s. But for the last 20 years, progress has been very modest, although now we're starting to see some things like PARP inhibitors. So we do have some promising therapeutics on the horizon. But even with all of that, 5-year survival is less than 50%. Right now, it's at about 48%. So the question is, why this poor survival? And it's believed to be because the vast majority of cases, about 75%, are diagnosed at these later stages 3 and 4, when survival is only about 30%. And the belief is if we could shift diagnosis from these late stages to these early stages, we can get survival to be over 80%. In a minute, we're going to talk about the validity of that belief. So just to step back for a second to clarify what I mean when I say ovarian cancer is epithelial ovarian cancer. And that accounts for about 90% of ovarian cancers. And I show here the 4 major histotypes, which, as you can see from the H&E stain slides on the left, are very morphologically distinct. But from the graphs on the right, you could also see that they have very different clinical presentations. Here we see the differences in stage at which they present, and this is outcomes, so very different. And the serous subtype here has the worst survival. It's also the most common. About 60 to 65% of women will be diagnosed with serous epithelial ovarian cancer. So now we know that ovarian cancer is really a group of different diseases. Where ovarian cancer originates, well, about 20 years ago, this was a very hotly debated topic. It was believed to be the ovarian surface epithelium, so that when a woman ovulated and there was damage to the surface epithelium of the ovary, an inclusion cyst would form and something would go wrong in there, and that was believed to be the genesis. But we now know with careful sectioning of both the ovaries and the fallopian tubes of BRCA carriers that a large majority of ovarian cancers actually arise in the fallopian tube. Now, BRCA carriers have about a 70% lifetime risk of developing ovarian cancer, and it's pretty much the serous subtype. And so with the advent of molecular markers, we now know that the different histotypes actually have different cellular origins. So the serous type, which is broken down into high and low grade as 2 different disease entities, arise from most likely the fallopian tube, although there's evidence that some actually arise from the ovarian surface epithelium. For clear cell and endometrioid, those are believed to arise from endometriosis. Endometriosis is now considered a precancerous state. Now, what causes a woman with endometriosis to develop cancer, ovarian cancer, we don't know, but some small percentage will go on to be diagnosed with clear cell or endometrioid ovarian cancer. And actually, that leads to how does this happen? The theory is possibly retrograde menstruation. But really what this tells us is that these subtypes of, quote, ovarian cancer likely have their origin in the endometrium and not the ovary. And then the 4th histotype that we'll talk about is mucinous carcinoma. There's very little known about that origin. So it's very important that when we study ovarian cancer, we just don't lump everyone together, but that we have a sample size that's large enough to enable us to look at least these 4 main histotypes. And there are other subtypes. And that's one of the powers of Consortia that we'll talk about. So continuing on, why this dismal outlook for women diagnosed with ovarian cancer? If you talk to women, they'll tell you that their symptoms were very vague and that it took them a while to get their diagnosis. And that was believed to be the reason why we see this late stage diagnosis. And again, we'll talk about that in a minute. If you take anything away from my talk today, it's that any woman with suspected ovarian cancer should get to a comprehensive cancer center like UAB or like the Hillman here, because about half the cases treated in the community don't get standard of care treatment. And after the skill of the surgeon, that's the #1 predictor of outcome. So it's pretty shocking that we have women today still not getting what we know is the best treatment. We also know that we don't have effective treatments for recurrent disease. So most women with ovarian cancer will recur within about a year and a half. And then they enter this cycle of they go into a brief remission and then a recurrence, and a remission and a recurrence. And once the woman's cancer becomes platinum resistant, we have no current treatment options. So as an epidemiologist, I hold that right now, early detection and prevention are critical for us to overcome this disease. So in terms of early detection, let's talk for a minute about screening. I mentioned that ovarian cancer is rare. The prevalence is about 1 in 2,500. And we know that a very rare disease for asymptomatic screening test is going to require a very sensitive and extremely specific test. And for ovarian cancer, just having a 75% sensitivity and over 99% specificity would give us a positive predictive value of 10%. So what does that mean in reality? So once a woman screens positive for ovarian cancer, the next step is actually surgery. And what this tells us is that for every 10 positive screens that go to surgery, we'll find one ovarian cancer. So that's a lot of intervention to find one cancer. So we really need to do better at early detection and prevention. So let's talk a little bit about screening and what we know from screening tests. I'm assuming many of you are probably very familiar with PLCO. I believe UAB was a site. But in this study, the intervention was, no intervention, just standard of care, which is you present with symptoms and then you go on for workup, versus the women who got an intervention had a baseline and annual transvaginal ultrasound and CA-125. So the only screening tools that we have currently are an imaging modality, which is transvaginal ultrasound, and a blood-based marker, which is CA-125. And neither alone satisfy the sensitivity and specificity requirements we talked about. But as you can see, so these women in the intervention were having annually both screens together. After 15 years of follow-up, as you can see from the graph, there were no differences between intervention and standard of care. So we saw no benefit from that type of screening. The other big screening trial was the UKC tox, and they had two interventions. One was transvaginal ultrasound alone, and the other one I found very interesting was to look at longitudinal changes in CA-125. So instead of having just a cutoff that would warrant further workup, they actually monitored a woman's CA-125 levels, and they had an algorithm that as it changed, and once the change reached a certain threshold, that would trigger further workup. And as you can see, though, after 16 years of follow-up, there were no difference among the three different arms. Interestingly, about 10% of the study population, there was about a 10% decrease in stage 3 and 4 disease shifted to stage 1, but yet we saw no benefit from this screening. And in fact, in the multimodal arm, which was the longitudinal change followed by a TVU if it got triggered, then followed by surgery, the shift was even more dramatic. It was about a 25% shift, and again, no difference. So that tells us that there's something more going on than just needing to shift the stage of diagnosis. So that didn't pan out. So we now know we still don't have any effective screening or early detection tests. So without a screening mechanism, we need to focus on prevention. Unfortunately, we really don't have much in terms of prevention beyond oral contraceptive use and removing a woman's ovaries. And that's because we really don't have many established modifiable factors. So these are the different epidemiologic factors that have been studied in ovarian cancer. Unfortunately, many of them, like age or BRCA status, are not modifiable. And something like an oophorectomy, which is the removal of a woman's ovaries, that has substantial health consequences. In particular, it immediately throws a woman into menopause, and it has a lot of consequences for things like cardiovascular risk. So we need to look at potential modifiable risk factors. And we know that there are 3 established ones, using oral contraceptives, bearing children, and breastfeeding. And We know that these have been consistently associated with reduced risk of ovarian cancer across all different study types and all different populations. We've seen about a 25 to 30% reduction in risk that increases the longer a woman is exposed to one of these behaviors, and more recent is more protective. And importantly, whatever protection a woman gets lasts for more than 30 years. So we also know that risk factor profiles vary by histological types, and this actually confirms what we've talked about, that ovarian cancer isn't a single disease, but different disease entities with different origins. So that opens the question as to what are the hypotheses to explain ovarian cancer's origin. So there are several, and most of them are based on the link between oral contraceptive use or bearing children and ovarian cancer. The most prevalent one in the literature has been around since 1971, and everyone still talks about it. It's suppressing ovulation. So the theory that was put out was that when you suppress ovulation due to oral contraceptives or bearing children, and now even potentially breastfeeding, you reduce the trauma from ovulation to the ovarian surface epithelial that could lead to this aberrant repair that we talked about. But that opens the question to what do we do about the fallopian tubes as well as the endometrium, and we'll talk about at least some of that in a minute. So there are a lot of different other theories related to hormones. Elevated gonadotropins is also one that's often cited in the literature. They're associated with increased estrogen production and epithelial cell proliferation. But elevated estrogens, and we'll talk about excess androgens and reduced progesterones, those have also been posited to play some sort of role. And then back in the 1990s, inflammation was hot in cancer origins. So there are also hypotheses that talk about these are all somehow related to inflammation. So that's the background. And now I'd like to look at some of the work I've done with students and looking at these hypotheses that we just talked about. So the first I want to talk about is the ovulation hypothesis. This is actually the first paper of my most recent PhD student, Xu Shen. She just defended last month, actually. And her grandmother actually died from ovarian cancer. And so she was very interested in prevention, and she became very interested in this incessant ovulation hypothesis. And so we wanted to look into what's the association between lifetime ovulatory years and ovarian cancer risk. In lifetime ovulatory years, we're going to define as menstrual span. And because I don't have a whiteboard and can't be drawing, I'm just going to use my hands. I'm going to use this to mean menstrual span. Okay, so the time between a woman's last menstrual period and her first menstrual period is going to be her menstrual span. And from that window, we're going to take away any years of anovulation due to pregnancy and oral contraceptive use in pregnancy. And we're going to define that reduced window to be lifetime ovulatory years. So the first thing I always have my students do is go to the literature. And so not surprising, because this hypothesis has been around now for 50 years, there were a number of papers, 21 to be precise, that reported effect sizes. And I thought, hey, let's do a meta-analysis. And it sounded so simple and straightforward at the time. And so what we did was first graph, these are the different papers. And actually there's more than 21 here because a few papers had different subpopulations. So there were 24 total from 21 different papers. And what we graphed was the estimated lifetime ovulatory years versus the adjusted odds ratio. And so as you can see, these graphs all go up and to the right, which says that more lifetime ovulatory years is associated with a greater odds ratio of ovarian cancer. Okay, so now there were some challenges in looking at these data. So first, the papers defined lifetime ovulatory years differently. And so that made direct comparisons challenging. And so that was something we wanted to think about. And then because we're epidemiologists, we all love to categorize continuous variables. The challenge, though, was that different papers chose different cut points. So what we really see graphed here is the middle of each of the different categories of each of the different papers. So we couldn't really do direct comparisons. We could only kind of look at that graph and make some conclusions. And then finally, important to me, is that because they had limited sample size, they really couldn't look at histotype analyses. Very rarely were they able to do that. And so we know that that's something that's important because we know that we're looking at different diseases. So what we decided to do was to compare however any paper defined their highest category of exposure to their lowest category of exposure. And that's what we graphed here. And then we Shushen created a pooled odds ratio and showed that the highest to lowest category was an odds ratio of about 2.26. So what this really just confirms to us is greater lifetime ovulatory years is associated with increased risk. What it doesn't give us the ability to do is to say, create a variable that we might want to put in a risk assessment model because we want to see if we could identify high-risk women and create interventions given the rarity of this disease. And I just want to say that there were 4 papers that looked at some of the histotypes, and we saw this result was consistent for the serous and the endometrioid, and it's questionable for the mucinous. Now, remember, I told you mucinous we know very little bit about, and we're going to talk in a few minutes that even the risk factor profile for mucinous is very different. So this suggested that maybe lifetime ovulatory years was not associated with mucinous tumors. But again, too small a sample size to really draw firm conclusions. So this left us some open questions that made the basis for Shushen's first dissertation paper. First, we wanted to know, could we provide a more precise estimate of the effect of lifetime ovulatory years to, for example, include in a risk assessment model? And then we wanted to know, because we saw different definitions of lifetime ovulatory years, how those definitions might affect our estimates. And then the third question was, how are the components that make up lifetime ovulatory years independently associated with ovarian cancer risk? And for me, this is the most interesting question, because if we can really understand that, then we can really get at the question of, is it ovulation suppression alone, or is there something else going on? And then finally, as always, very interested in the histotype specificity. So in trying to answer this question, we went to OCAC, the Ovarian Cancer Association Consortium, which was formed in 2005. Hope, one of my studies, was a founding member. We began as 19 case control studies looking at genetic risk factors. That's why we're the Association Consortium. We now have over 70 studies on 5 continents, and our studies range from classical epidemiology to more molecular epitype work. And one of the beauties of OCAC is we have this available core epidemiologic data set, and as well as a clinical data set that have been cleaned and harmonized across all the studies so that we can just do some large-scale pooled analyses. So in the OCAC core data set, these were the variables that we found. So they were already cleaned and harmonized. And we found 25 case control studies that had complete data from 4 continents and 9 different countries. The case control studies were all different types, like hospital and population-based or registry-based. And we had a total of over 21,000 cases and 26,000 controls. So again, these are the variables that are going to go in our definition of lifetime ovulatory years. And let's see how we put them together. So we came up with 15 different ways to put them together, and then we did some sensitivity analysis. So it's more than 15, but these were the 15 main ways. So remember, as I said, we started with menstrual span, which is the last age at last menstrual period minus the age at menarche. And then we want to take away periods of anovulation. So the first one was for duration of pregnancy. Now, each of these columns is going to be a different way that we subtracted duration of pregnancy. So here, for all the pregnancies, we subtracted 3/4 of a year. So column A will always be that. Column B, we restricted to only full-term pregnancies and subtracted 3/4 of a year. In column C, we included all pregnancies, but we weighted them different. A full-term pregnancy was 3/4 of a year, and a non-full-term pregnancy was only a quarter of a year, 3 months, so 9 months versus 3 months. And then OCAC had this clean variable called months of pregnancy, so we used that just as a check, but I wasn't as enthusiastic about using that, but we did. Okay, so then after we took away duration of pregnancy, we took away duration of oral contraceptive use. So all of those algorithms have the same metric for duration of contraceptive use, but just a different definition of pregnancy. And then we added in duration of breastfeeding. And now there were a handful of studies in OCAC that had average cycle length. And so for those, we also we did analysis based on some of those, but it was small. So I wanted to just use those as just kind of a check to see what was going on. And so these are our results, our main results. So our first question was, what was the association per year of increase in lifetime ovulatory years? And we know with the sample size, the size we had, we were able to provide a more precise estimate. And then we wanted to look at the effects of the definition of lifetime ovulatory years. And that's the busyness of this slide. So these are the 4 different ways pregnancy was defined. And in this group, we only subtracted pregnancy. As you can see, the definition of pregnancy made really no difference. Here we put in oral contraceptive use, and as you can see, it did make a modest impact. Here we put in breastfeeding, and we saw a little change, but not much. And then cycle length actually did make a difference, but again, we didn't have very many studies, so it would be nice to have more studies with average cycle length to include, but we did see that it did make somewhat of a difference. And then these 2 columns, these are our adjusted odds ratios where we included other confounders. And for any confounder that had an unknown, we used a category, unknown. And then what I had Shushan do was to do some imputation just to see what does the impact of an unknown variable have on our estimates. And as you can see, comparing this column to this column, with the exception of breastfeeding, it really had no impact. So those are our first 2 questions. Our third question was looking at the association of the components of lifetime ovulatory years with ovarian cancer risk. So now this is one single logistic regression model where we put our confounders in, and then instead of putting lifetime ovulatory years, we put each of the components. And as you can see, with the exception of age at menarche, all of the components are significantly associated with ovarian cancer risk. And that's not surprising, we would expect that. Okay, but what we then wanted to do was to understand whether these relationships were based on ovulation suppression alone. Okay, so if ovulation suppression alone is the mechanism for a mechanism of action of each of these components, then 1 year of suppressing ovulation, regardless of the source of that ovulation suppression, would be the same, would have the same effect on our logit, right? So what I'm saying is, if this is our menstrual span after we've taken away an ovulation, if we, for example, add a year because a woman has 1 more year, a later year of menopause, that would be the same as 1 more year because she had her age at first menarche earlier, or she had 1 less year of oral contraceptive use. So for each unit of exposure, if the component, for each unit of exposure for each component, if it's 1 year, then we would anticipate that our beta coefficients would be the same, okay? So what we did was create a theoretical model. And we said, let's define the beta coefficient of our age at last menstrual period to be 1. Okay, so if X1 is age at last menstrual period, beta 1 is going to be 1. So if we add 1 year here, that's 1, okay? So since adding 1 year at age at menarche would actually take away a year, that theoretical beta coefficient would be -1. And the same for 1 more year of oral contraceptive use. For pregnancy, our theoretical for a full term would be -0.75 because 9 months is 3/4 of a year. And for a non-full term, we said -0.25 because we estimated 3 months. And then for breastfeeding, again, just -1. Okay, so these are our theoretical beta coefficients. So now we're going to look at our actual logistic regression model, and we're going to look at our actual beta coefficients. But what we're going to do is we're going to normalize them to the beta coefficient of age at last menstrual period, so we can compare how close our actual estimates are to our theoretical, okay? So what we're going to do is if beta 1 is the coefficient for age at last menstrual period, we divided all our beta coefficients by beta 1. That would set this coefficient to 1, just like our theoretical model. And then this will tell us our relative beta coefficients relative to age at last menstrual period. And we can compare them to our theoretical estimates to see how far off we are. Okay. So as you can see, these are our relative ones, okay? Age at last menstrual period is 1, and these are the other ones. And as you can see, they vary, these 4 vary significantly from our theoretical estimates. For oral contraceptive use, our relative coefficient was 3 times what we would have anticipated. And then for pregnancy, whether or not it was a non-full term or a full term, it's about 7 times. And for 1 year of breastfeeding, it was about 5.5 times. And we did a lot with breastfeeding because I had a lot of questions about breastfeeding to show whether we used 6 months or capped it. We still found a difference that breastfeeding's relative coefficients were greater than their estimated. We would have theoretical rather are expected. So this tells us that given that our relative coefficients are greater than our theoretical coefficients, that the effects of these exposures has to be beyond ovulation suppression, right? Because we define them in a way that it should have been the same if it's only suppressing ovulation that's going on. And then secondly, what we find is the effects of pregnancy, we said was 7 times compared to only 3 times for oral contraceptive use. So the effects of pregnancy, suppressing ovulation due to pregnancy is greater than that of ovulation of oral contraceptive use. Okay, so our final question was whether these associations differed by histotypes, and by now I hope you would have all guessed the answer to that would be yes, because we know they're different diseases. So if we look at lifetime ovulatory years as a continuous variable, we see differences. In fact, we see that for the mucinous type in particular, that's that one that I keep saying is out in left field, lifetime ovulatory years was not significantly associated with mucinous tumors, nor was it associated with low-grade serous. And then if we look specifically at mucinous tumors, we see that the associations in fact differ. In fact, age at last menstrual period seems to have no effect, which is in contrast to the other main histotypes. But age at menarche is positively associated, which again, it's not associated with the other main histotypes. So this just confirms to us that mucinous has a different pattern of risk factors than the other histotypes. And even though we see these positive associations between the components of lifetime ovulatory years, we saw no relationship with lifetime ovulatory years. So this tells us again that there's something else going on beyond ovulation suppression, specifically for mucinous tumors. However, when we look at also the other histotypes, and the one I want to call your attention to, because I found this really interesting, was the high-grade serous. Now remember, that's the most common. That's believed to arise predominantly in the fallopian tubes. But as you can see, with the exception of breastfeeding, the actual estimates were very close to the theoretical estimates here, which is, again, very interesting because, again, high-grade serous is not believed to, most people believe it does not arise in the ovary, but in the fallopian tubes. So that tells us that definitely there's something going on potentially with ovulation that doesn't have anything to do with the ovarian surface epithelium. So I spent a lot of time researching that because I became very interested, and there's a lot of emerging data now that is implicating potentially follicular fluid. So this is the ovary, this is the fallopian tube. When a woman ovulates and the egg goes down, follicular fluid is also released, and that does reach the fimbriated end of the fallopian tube. And if you look at the literature, it's very fascinating. When they have cultured normal fallopian tube with follicular fluid, they've found aberrant P53 expression, which is a hallmark of high-grade serous cancers. And they've also shown that follicular fluid has both mutagenic and tumorigenic effects on the fallopian tube. So that implicates that it might be the follicular fluid that's the operative agent, at least for high-grade serous. Okay, so quickly in answering our question, we found we can get more precise estimates. We found that the definitions of LOY do affect somewhat, but for the most part, we saw a consistency of our findings. I found the most interesting thing that resulted from this work is that the impact of the components is beyond ovulation suppression. And that we see that this, number 3, is, we've observed all these associations, in fact, in a very histotype specific way. Okay, so now we know that there's something above and beyond ovulation that has to explain the association between these reproductive factors and ovarian cancers. So other factors that are associated with these components, such as hormonal alterations, are likely at work. So let's look at the hormone hypothesis. So one factor that I want to focus on is pregnancy. So recall that the impact of pregnancy on ovarian cancer risk was about 7 times what was expected from ovulation suppression alone, although that wasn't true for high-grade serous. And one factor related to pregnancy that I became interested in 20 years ago was offspring sex. So together with my students, I did look at this relationship in 2 different populations. One was conducted in Eastern Pennsylvania, the SHARE study. That was a data set I inherited. And that study was done back in the early 1990s. And the other was done here in Western Pennsylvania in the early 2000s. And this first analysis, the SHARE analysis, was done by my very first graduate student, Gretchen Gierak, who now heads the Integrative Epidemiology Tumor Branch at NCI. And the second paper was Xu Shen's first paper that she did when she joined my group. And so for me, I want to take a step out of the science and tell you that I think it's really important that students begin to learn about epi by actually doing epi and getting their hands in there as early on as possible. So I like to start students off with doing some analyses in existing data sets and reproducing analyses. It's a great way for the student not only to learn epidemiology, but we get to learn about each other and how to work together. And so what we found in both populations was that male offspring is associated with reduced ovarian cancer risk. Now, this is kind of perplexing because pregnancy, regardless of fetal sex, will equally affect ovulation, right? So we shouldn't have seen a difference between male and female pregnancies. And they also equally affect gonadotropin. So we would expect that there might not be differences in hormones. So at least something else might be going on here. So let me take a step back and tell you why in 2005 I had this idea to do this, which was, like most of my work, driven by personal experience. And at the time, there was a lot of focus on the roles of hormones in ovarian cancer, especially androgens, sex steroid hormones. And this was a hypothesis that was put forth in, I think, 1999 by Harvey Risch, 98 rather. And so he hypothesized that androgen exposure was associated with an increased risk, while progesterone or progestin exposure would be associated with a reduced risk. So our group was using the SHARE data set to do all sorts of analyses to look at that hypothesis. And I just show one where we looked at treatment of endometriosis, with an androgenic versus a non-androgenic agent. And we showed that the androgenic agent was associated with an increased risk of ovarian cancer and not the non-androgenic agent. That was what was going on at the time. And I reflected on my pregnancies and recalled that with my firstborn, which was my son, my face broke out, which is a sign of excess androgens. And with my second pregnancy, which was my daughter, I had this beautifully glowing skin. And so I went to the literature and sure enough, learned that there is some hormonal spillage into maternal circulation. In fact, male pregnancies are associated with lower estradiol, which makes sense, and lower HCG, but higher progesterone and testosterone, which is also makes, well, the testosterone makes sense to me. But how this fit in with my observation, I really don't know. Our observation of reduced risk of ovarian cancer with male offspring, because I would have expected that we would have either had a null finding due to the progesterone or an increased risk due to the testosterone. And so I kind of questioned this, and then I started thinking, well, would that little bit of extra hormone exposure really make a difference to a woman's risk of ovarian cancer? And I felt the argument was weak. But nonetheless, I continued on and made the argument in my papers that pregnancy conditions vary by fetal sex, and that these conditions that vary by the gender of the baby you're carrying are associated with future mother health outcomes. So I made the argument that fetal sex-based differences can have long-term health consequences for the mother. And so there is some biological plausibility for our observation, albeit weak. And it bothered me for a long time. And I thought, well, maybe it's just a spurious finding. And then this paper came out and I thought, this is it. This explains everything. It was this paper here, and it was on something called male origin microchimerism. Microchimerism is the presence of genetically distinct cells within an individual. And it's actually a common phenomena. And about 70% of healthy women carry this male origin microchimerism, with the most common source being pregnancy. And from the literature, we know that this microchimerism really persists for many, many years after pregnancy. And even though it's present in very small quantities, it actually has a remarkable impact on women's health. For example, women who are microchimeric positive compared to women who are microchimeric negative have a 70% reduced risk of breast cancer pre and postmenopausal, and also reduced risk of endometrial cancer. And then earlier this year, this interesting article came out in the American Journal of Epidemiology on showing that microchimeric positive women have a 56% reduced risk of ischemic heart disease. And so this paper on ovarian cancer also showed a 56% decrease in ovarian cancer risk with this male origin microchimerism. And I thought, well, that's it. That explains our observation, why carrying a male offspring would be associated with a reduced risk of ovarian cancer. And I was so proud of this finding. And so I went to OCAC and I said, we have to do this in all our OCAC studies. Now, I told you we have a core epi data set that's cleaned, but offspring sex was not one of the variables in there. And so we had to go to each individual study, and we found 12 case control studies that did have offspring sex. And we wanted to confirm this great finding in OCAC. And then I wanted to use the GWAS data from OCAC to look at this genetically and confirm it that way, confirm our epi study. And so here are our findings, and let me save you the trouble of having to squint and read. We actually found no difference in the risk of ovarian cancer in general between having a male offspring and ovarian cancer risk. However, we found an increased risk with this mucinous subtype. So we found a completely different finding than I had found in previous work. And remember, mucinous has this very different risk factor profile. Well, here's another way in which it's very different. So I can only say that the question of offspring sex and ovarian cancer remains an enigma to me, because we don't see any impact in this pooled analysis, which contradicts the genetic work that came out of Denmark, And I'm still waiting to be able to do this in OCAC because there are now issues with genetic data from Europe going to the US. So hopefully we'll resolve that at some point. We'll be able to look at it. But for the most part, no finding there. So that was disappointing, but that's the nature of research. So this brings me to the last 5 minutes of my talk, where I want to just talk a little bit about molecular epi, since I love doing molecular epi. And I'm going to talk about redefining, using molecular epi to redefine some of these rare subtypes. And in particular, I want to talk about clear cell carcinoma. So remember, we said that there were all these different disease origins. Clear cell carcinoma we trace back to endometriosis and possibly the endometrium. It's very rare. It represents only about 10% of ovarian cancers. And because of that, there haven't been these large TCGA-like studies of clear cell carcinoma looking at these molecular markers. Clear cell carcinoma has very poor survival, mostly because it's platinum resistant. And in contrast to the high-grade serous that I talked about, it's actually P53 wild type, but it does harbor two very consistent somatic mutations in PIK3CA and ARID1A1, which are actually frequently found in endometrial cancer. And so that, again, implicates the endometrium as a possible origin for this type of, quote, ovarian cancer. So because of the rarity, we formed our own consortia, and we pooled expertise from different sources for the leadership team. Ellen Good at Mayo is our genetic epidemiologist. Kelly Bolton was at Sloan, now she's at WashU. She's our medical oncologist. Julie Cunningham is our amazing multi-omic large data set analyst, which is a rare find to find someone who could do that. And then our group was the data coordinating center. We obtained all the medical record data on these women, and we cleaned them and put them in a harmonized data set. And this is another place where I love working with students because it's a big win. We get a clean data set, but I really love the conversations that we have about understanding clinical data and how we use clinical data in epidemiology, and how messy clinical data can be, and how do we handle messy data, and how do we handle missing data. So it's a really great topic for conversation in our meetings. Okay, because of the time, I'm just going to whip through these really fast. So what we did was we got a fresh frozen tumor on these 271 women, and we did methylation and as well as gene expression. And so from our methylation, we found two clusters that were defined by 977 genes. You can see this cluster, here's one cluster and here's the other cluster. We saw that these clusters had very different clinical presentations and especially outcomes. So cluster one is going to be the one here on the bottom, the blue, that has worse outcome, poorer survival. And it also had also different worse clinical presentation. But more importantly, it had very different tumor characteristics. It had more likely to have aberrant P53 expression and P53 mutations. And remember, I told you that's not a hallmark of clear cell carcinoma. That's a hallmark of high-grade serous. And we did have pathological review to ensure that these were not high-grade serous. They were, in fact, clear cell on a pathologic review. And then our second cluster harbored those two mutations that I told you were hallmarks of clear cell carcinoma. So this is the more classical clear cell. What we found a rarer, about half of the women were in cluster one. That is very different from what we anticipated. Then we did our gene expression analysis, and we found 369 differentially expressed genes between the two clusters. And we performed gene set enrichment analysis and found that these genes bind into 9 of the 50 hallmark gene sets. 6 of these gene sets are related in some way to the immune system, including interferon gamma and alpha response and inflammation. And then in further analyzing our RNA expression, we found 23 genes that had significantly different methylation expression relationships by clusters. In other words, a methylation cluster gene expression interaction. So along the x-axis here, we have the beta values for the CpGs, and along the y-axis, we have our gene expression level. And as you can see, cluster 1, which is the black line, and cluster 2, the lighter line, you can see the interactions very clearly in these 4 genes. Almost all the 23 genes were somehow related to the immune system and inflammation. So to summarize, among the 271 cancers, clear cell cancers, we found 2 clusters using our DNA methylation that had very different gene expression patterns, clinical courses, and tumor mutation signatures. And these 2 broad groups differentiated based on somehow immune-related factors. And so that's a new area to investigate potential therapeutics. So thank you for your time today. That brings me to the end. I want to thank everyone that's been involved in my work over the years and in this work in particular, as well as the different funding sources. And I'd just like to close by acknowledging my 30-plus year research and support collaboration with my husband. Here he is. He's currently now a professor of computer science here in Pittsburgh. We've undertaken 2 very intense research projects. This little guy here, this is him in 2018. He's going to turn 23 in January. He's out working now in Chicago, although he's commuting back and forth to London. He just got home yesterday from London, so he'll be going back in December. And then my pumpkin here just turned 20 last month. She did learn how to make it on to 2 from all 4s, and she's a 2nd-year student now up in Boston at MIT, much to the dismay of her lifelong education and faculty father from Carnegie Mellon, and my son also went to Carnegie Mellon. I'm okay with it as long as she just doesn't go over to the Patriots.